Nobody who has ever failed a diet failed because they lacked willpower. That framing is not just wrong. It is actively damaging to the people who believe it. The real reason dietary changes don't move the needle for so many people has nothing to do with what they ate between Monday and Friday. It has everything to do with what their body is doing with sugar between 10 troughs at night and 6 troughs in the morning. This is a window that most nutritional advice completely ignores. Here is the uncomfortable truth. Every night while you sleep, your liver is either converting excess blood glucose into stored fat, or it is clearing that glucose efficiently and preparing your metabolism for the next day. Which of those two things happens depends less on your diet than on a specific biological environment created in the hours before bed. This is an environment you can measurably shift with a single drink. Not a supplement, not a meal replacement. A drink made from ingredients that cost pennies, and that clinical research has found to meaningfully alter the way your body handles blood sugar during your overnight fast. This discussion is not about diets. It is about understanding the mechanism your body actually uses to regulate blood glucose overnight, and then giving it the one thing that allows that mechanism to work properly. Having spent years in emergency medicine, I have observed people arrive in crisis from metabolic conditions that were present and progressing long before they sought care. The most common thread is not ignorance. Most of these patients had been told to eat better and exercise more. The thread is a fundamental misunderstanding of when the body is most vulnerable to glucose dysregulation and what interventions actually work at that specific time. That is what needs to be addressed today, because preventing metabolic disease starts long before someone needs emergency care. Most of the information circulating online about blood sugar and diet misses the window that matters most. To understand why this drink works, you first need to understand what is happening inside you while you sleep, because this is the part that no dietary advice talks about. When you finish your last meal of the day, your blood glucose rises. Your pancreas releases insulin, and your body begins the process of either storing or clearing that glucose. Under healthy conditions, muscle tissue and the liver absorb the bulk of it. Muscle acts like a large sponge. The bigger and more metabolically active the sponge, the more glucose it can pull from the bloodstream before insulin even has to do much work. But here's where it gets interesting. Once your muscles are reasonably saturated with glycogen, which is the stored form of glucose, the liver becomes the primary processor. And the liver, unlike muscle, has a much more problematic fallback plan when glucose keeps arriving in excess. It converts that glucose into fat. Specifically, it converts it into triglycerides through a process called de novo lipogenesis. This is not some marginal side reaction. A study published in the Journal of Clinical Investigation found that in individuals with elevated blood sugar, de novo lipogenesis can account for a substantial proportion of new liver fat accumulation, contributing directly to non-alcoholic fatty liver disease, insulin resistance, and the kind of chronic metabolic dysfunction that plays out over years before anyone notices. Now, here is the part that is relevant to what we are discussing today. Your liver does not behave the same way during the day as it does at night. The liver runs on a circadian clock, a 24-hour biological rhythm governed by clock genes inside its cells. During daylight hours, the liver is primed for active glucose processing. At night, it shifts into a different mode. It reduces active glucose uptake. It increases glucose output through a process called hepatic glucose production, and it initiates cellular repair. The problem is that if blood glucose and insulin remain elevated when you go to bed because your last meal was high in refined carbohydrates, or because you ate too close to bedtime, you are essentially forcing the liver to continue metabolic processing at the exact time its circadian programming is telling it to wind down. The result is inefficient glucose clearance, elevated fasting blood sugar in the morning, and over time, a progressive increase in insulin resistance. This is what diets fail to address. Changing what you eat without changing when you eat, and without supporting the overnight metabolic environment, addresses only part of the picture. The drink I am going to describe works precisely on this overnight window, and the research behind it is more substantial than most people realize. The drink is a warm solution of diluted raw apple cider vinegar and Ceylon cinnamon, taken approximately 30 minutes before bed. That is the whole thing. No proprietary blend, no subscription required, nothing that requires you to visit a specialist store. These are two ingredients that have been studied independently for their effects on blood glucose, and that when combined appear to work through complementary mechanisms. Before explaining the science, it is important to address the obvious skepticism. The wellness industry has spent years co-opting apple cider vinegar and cinnamon as cure-all remedies, which has caused many people to dismiss both as pseudoscience. But the research that exists here is not testimonial. It is peer-reviewed, mechanistic, and increasingly well-replicated. The reason these ingredients are so often misrepresented is that people describe the outcomes without explaining the mechanism. Without the mechanism, it sounds like magic. With the mechanism, it makes complete physiological sense. The active compound in apple cider vinegar that drives the metabolic effect is acetic acid. When you consume diluted acetic acid, it is absorbed rapidly from the stomach and small intestine and carried to the liver, where it does something remarkable at the cellular level. Inside liver cells, acetic acid is converted to acetate, which then gets phosphorylated into acetyl-CoA. In doing so, it transiently shifts the ratio of AMP to ATP inside those cells. This matters enormously because that ratio shift activates an enzyme called AMP-activated protein kinase, or AMPK. Think of AMPK as the body's master metabolic switch. It is the enzyme that essentially tells cells to stop storing energy and start burning it. AMPK activation suppresses a process called hepatic glucose production, which is the liver's tendency to release stored glucose back into the bloodstream overnight. It also enhances glucose uptake in peripheral tissues and improves the sensitivity of insulin receptors. A landmark study published in Diabetes Care tested the effect of vinegar ingestion at bedtime in patients with type 2 diabetes. They found that fasting blood glucose levels in the morning were reduced by an average of 4 to 6% compared to placebo. This was a clinically meaningful reduction achieved by a single bedtime dose of vinegar and water. The research team attributed this specifically to the suppression of hepatic glucose production through the acetate mechanism. Practically, this means that instead of your liver quietly releasing glucose into your bloodstream throughout the night, elevating your fasting glucose, and forcing your body to respond with insulin even while you sleep, the acetic acid partially breaks that process. You wake up with lower blood glucose, lower morning insulin, and a metabolic starting position that is genuinely better for the rest of the day. Think of it less as flushing sugar out, and more as closing a gate that would otherwise allow glucose to keep circulating when your body has no constructive use for it. The second ingredient, cinnamon, works through a different but complementary pathway, and the distinction between types of cinnamon matters here clinically. There are two main varieties of cinnamon available commercially. Cassia cinnamon, the variety most commonly sold in supermarkets, contains high concentrations of a compound called coumarin. Coumarin is hepatotoxic at regular doses, meaning it causes liver damage over time. This is not hypothetical. The European Food Safety Authority has established tolerable daily intake limits for coumarin precisely because of this risk. Ceylon cinnamon, sometimes called true cinnamon, contains trace amounts of coumarin by comparison. It is low enough to be considered safe for regular daily use. If you are going to add cinnamon to a nightly drink, Ceylon cinnamon is the only version that makes sense. It is usually slightly lighter in color, has a more delicate flavor, and is available in most health food stores or online. This is a real safety consideration. Now, for the mechanism. Ceylon cinnamon contains bioactive compounds, primarily cinnamaldehyde and A-type proanthocyanidins, that interact with insulin signaling at the receptor level. Insulin works by binding to receptors on the surface of cells, which then activate a cascade of internal signals that tell the cell to import glucose from the bloodstream. In insulin-resistant states, those receptors have become desensitized. They respond sluggishly, the cascade runs poorly, and glucose accumulates in the blood, even in the presence of adequate insulin. Cinnamaldehyde and the proanthocyanidins in Ceylon cinnamon appear to activate insulin receptor kinase, the enzyme that initiates that internal signaling cascade, independently of insulin binding itself. They essentially amplify the downstream signal even when the receptor is partially desensitized. Research published in the Annals of Family Medicine analyzed a series of controlled trials and found that cinnamon supplementation produced meaningful reductions in fasting blood glucose and triglycerides across multiple studies. A later analysis in the Journal of Medicinal Food found that this effect was most pronounced in individuals with prediabetes or early insulin resistance. This is precisely the population where overnight glucose dysregulation is doing the most cumulative damage. The practical implication is this: while the apple cider vinegar is suppressing the liver's overnight glucose output, the cinnamon is simultaneously improving the ability of cells to respond to whatever insulin is circulating. It is a two-point intervention on the same problem from two different angles. The third mechanism is one that the research on both these ingredients is still unpacking, but the direction of evidence is consistent enough to be worth understanding. Both apple cider vinegar and Ceylon cinnamon appear to influence the composition and activity of the gut microbiome in ways that are relevant to overnight glucose metabolism. Bacterial populations continue their metabolic activity throughout the night, and some of those bacteria produce short-chain fatty acids, particularly butyrate and propionate, that have direct effects on glucose regulation. Butyrate produced by gut bacteria serves as a preferred fuel source for the cells lining your colon, but it also enters systemic circulation and activates GPR41 and GPR43, G-protein-coupled receptors that stimulate incretin hormone release and enhance insulin sensitivity. In straightforward terms, certain gut bacteria produce compounds overnight that help your body manage blood sugar better. Acetic acid, which is effectively what apple cider vinegar is itself a short-chain fatty acid. Research in cell host and microbe has demonstrated that acetate produced either by bacteria or consumed directly influences the relative abundance of beneficial bacterial populations, specifically reducing the growth of certain pathogenic species while supporting Akkermansia mucinifila, a bacterium consistently associated with better metabolic health and improved glucose tolerance in multiple human studies. Cinnamon polyphenols have been shown in separate research to increase microbial diversity and favor butyrate-producing species. What this means at a practical level is that this drink does not only work acutely on the night you take it. Regular use appears to gradually shift the gut environment in a direction that makes overnight glucose regulation progressively easier. You are not just managing a symptom each night. You are potentially changing the biological substrate that determines how well your body handles glucose across every overnight fast. That distinction matters a great deal from a longevity standpoint. The preparation is straightforward, but several common mistakes significantly reduce either the effectiveness or the safety of this drink. Start with warm water, not hot. The temperature matters for two reasons. Hot water above approximately 60 degrees C can degrade some of the enzymatic and probiotic components of raw, unfiltered apple cider vinegar, the cloudy material at the bottom of the bottle called the mother, which contains much of the biologically active material. Warm water also makes the drink considerably more palatable and helps with absorption. A standard mug of warm water is the right vessel. Into that water, add 1 to 2 TBSP of raw, unfiltered apple cider vinegar. The dose that appears in clinical studies typically falls in this range. Going above 2 TBSP offers no additional benefit documented in the research and significantly increases the risk of esophageal irritation and dental enamel erosion. The dilution in a full mug of water brings the acidity to a level that is safe for regular use. Do not drink this undiluted, and do not drink it through your teeth. Using a straw keeps the acetic acid away from enamel, which is a sensible precaution. Add half a teaspoon of Ceylon cinnamon, not cassia. As mentioned, the coumarin content makes cassia inappropriate for daily use. Half a teaspoon falls within the dose ranges used in the cinnamon research, showing glucose benefits without approaching any threshold of concern for coumarin-free Ceylon cinnamon. You can add a small amount of lemon juice, a squeeze, which adds vitamin C, supports liver function overnight, and makes the drink genuinely more pleasant to take. No sweetener. Adding sugar or honey defeats the purpose at a mechanistic level because you would be re-elevating the glucose that the drink is meant to help clear. Timing is important. Take this drink approximately 30 minutes before bed, not immediately before lying down. The positioning of the acetic acid delivery relative to overnight hepatic glucose production matters. The studies showing reductions in fasting morning glucose consistently used a pre-bed or late evening consumption window. If you have been diagnosed with gastroparesis, peptic ulcers, or chronic acid reflux, speak with your physician before adding this regularly. The acidity is safe for most people at this dilution, but is contraindicated in certain gastrointestinal conditions. One final practical note. If you are currently on any medication that affects blood glucose, particularly insulin or sulfonylureas, this drink can have an additive lowering effect. That is not necessarily dangerous, but it means your existing dosing may need reassessment if you start seeing lower morning glucose readings. Please discuss that with the doctor who manages your medication before making a change. It is important to be direct about something. You will not wake up tomorrow morning with dramatically different blood glucose after one drink. That is not how physiological change works. What the research shows is an acute effect, a measurable reduction in fasting morning glucose on the nights you take the drink, but the magnitude of that effect is modest on a single occasion. In the studies, the reduction in fasting glucose was clinically meaningful, but represented a shift of roughly 4 to 6% from baseline. If your fasting glucose normally sits at around 100 milliliters per deciliter, you might see it at 94 or 95 the morning after. That matters, but it is not a dramatic transformation you will perceive viscerally. What accumulates over weeks and months is more significant. The mechanisms described, such as improved insulin receptor sensitivity, hepatic glucose suppression, and gradual shifts in gut microbiome composition, are cumulative processes. Research examining cinnamon supplementation over 12 weeks consistently shows larger effects than single-dose studies. The gut microbiome takes time to shift in response to regular dietary inputs. Insulin receptor sensitivity improves gradually as the signaling environment changes. The honest answer to when you will notice a difference is probably 2 to 4 weeks of consistent use before the downstream effects on energy, morning glucose readings, and appetite regulation start to compound in a way that is perceptible. From a longevity perspective, that timeline is completely irrelevant. The chronic conditions most directly linked to impaired overnight glucose metabolism, such as insulin resistance, type 2 diabetes, non-alcoholic fatty liver disease, and cardiovascular disease develop over years and decades. Interventions that incrementally improve the overnight metabolic environment, night after night for years, are precisely the category of low-cost, low-risk habit that epidemiological research consistently associates with meaningfully better long-term outcomes. There is one thing that reliably cancels the overnight glucose-regulating effects of this drink. And it is exactly the kind of error that is easy to make because it seems harmless. Eating refined carbohydrates or sugary foods in the 30 to 60 minutes before taking the drink. This sounds obvious, but the scenario seen most often is someone adding this drink to an existing routine that includes a late-night snack, a biscuit with the evening tea, a small dessert, or a piece of toast. The drink is not magic. It cannot meaningfully suppress hepatic glucose production when the liver is already dealing with a fresh glucose load from a pre-bed carbohydrate hit. The mechanisms described work optimally when blood glucose is declining and the body is transitioning into its overnight fasting state, not when it is managing active glucose absorption from the gut simultaneously. The simple rule: last food 2 to 3 hours before bed, drink 30 minutes before bed. That sequencing is not arbitrary. It aligns the drink's peak metabolic effect delivered through the AMPK activation and cinnamon receptor sensitization with the window when hepatic glucose production would otherwise ramp up unchecked. Compress that window with a late-night snack, and you are essentially asking two things to happen at once that work against each other. The second mistake is inconsistency. People take the drink for a few nights, notice no dramatic effect, and stop. The gut microbiome component of this intervention is particularly dependent on regularity. Sporadic input does not produce the gradual shifts in bacterial population that drive the longer-term insulin sensitivity improvements. Think of it less like a painkiller, where a single dose produces a single acute effect, and more like exercise, where the benefits depend on repeated, consistent stimulus over time. Everything covered today comes back to one idea that is genuinely underappreciated in the conversation about metabolic health. The hours between when you stop eating and when you wake up are not metabolically neutral. Your liver does not clock off. Hepatic glucose production continues. Insulin signaling either functions well or it does not. And the downstream consequences of getting that overnight window wrong, subtly, gradually, over years, are the same consequences that show up in metabolic disease patterns. This drink is not a cure. It is not a replacement for a well-structured diet, for regular physical activity, or for maintaining muscle mass as you age. What it is a scientifically coherent, low-cost, low-risk tool that addresses a specific and real physiological window that most health advice completely ignores. The mechanisms are real. The research is peer-reviewed and replicable. And the barrier to trying it is essentially zero. Your metabolism does not operate on a diet schedule. It operates around the clock, and the decisions made in the quiet window between your last meal and your first morning breath are quietly adding up, for better or worse, every single day of your life. The question is simply whether you give your body what it needs during that window to work correctly. If this explanation of the hidden biological mechanisms of your body has changed the way you think about your nightly routine, please subscribe to our channel and give this video a thumbs up. We are dedicated to uncovering the metabolic truths that help you take control of your health. Let's build a better future together. Our next deep dive into the body's hidden treasures will be opening soon.